My name is Joanna Wu. I am a TCBS graduate student at the University of Hawaii at Hilo. I am also a student with EPSCoR Cyber Team, and today I'm going to be talking about my research on native and non-native frugivore movement patterns and their implications for seed dispersal in Hawaii. This research is done in conjunction with my advisors Donna Delpart and Patrick Hart. So to start off, frugivore movements greatly influence seed dispersal, as when birds travel and go around and poop, they disperse seeds to places that plants can't normally disperse their seeds to. In Hawaii, this is especially important because half of all the native plants are bird dispersed, owing much to its isolation in the Pacific Ocean. Many of the plants were brought here initially by birds, and many have retained that mechanism of dispersal by birds. Historically, there were 5 species of frugivorous thrushes present across all the islands, but now, unfortunately, all of them have gone extinct or become critically endangered, except for the oma'o, which thrives on the island of Hawaii. Its range has been much reduced from its previous range. At the same time, many species of birds and plants have been introduced to the Hawaiian Islands, and of those birds, the most common and widespread is the Japanese white-eye. The white-eye not only eats fruits, but is opportunistic and eats all kinds of insects, nectar, and other types of foods. However, not much is known about its behavior and ecology, despite the fact that it's so common in Hawaii. We don't know about its roles in the ecosystem, such as competition with native species, or whether it could substitute for native species in places that they've been extirpated. I'm interested in looking at the roles of the Japanese white-eye in terms of seed dispersal compared to the oma'o. So looking at their movements and also what they eat, how they differ. My study system is a network of 34 kipuka on the Saddle Road area on the Big Island of Hawaii. It's about 1,500 to 1,800 meters, so it's a mid-elevation mesic site, and each kipuka is outlined in white. And a kipuka is literally a tree hole in Hawaiian where lava flowed down the hill and carved around a patch of previously contiguous forest. So taking you down to the ground, the matrix, which is the area surrounding the kipuka, is very young. It's from 1855 and 1880 lava flows off of Mauna Loa. So as you can see, there's not much vegetation colonizing the matrix yet. When you go inside one of those kipuka, which you can see in the background in the first photo there, it's a close canopy old growth forest, and it's one of the areas on the island that's got the best track of native forest. So the objectives of my study are to, 1, determine the spatial extent of seed dispersal of the 2 species and compare how they use this fragmented landscape. For example, are they moving between kipuka that are close to each other? What about ones that are farther apart? And is there a difference in movement patterns between the oma'o and the Japanese white-eye? Next, I want to determine just how frugivorous they are. So how much fruit are they actually eating versus insect and other foods? So to conduct the first part of my study, I used the method of radiotelemetry, where I attach radio transmitters to my 2 species of birds, the oma'o and the white-eye, and then I track them using 2 observers to intersect the bearings and get a location fix on the bird. And so we did this over a period of 1.5 years and looked at the distance that the birds travel during the gut passage time. For the oma'o, I used a gut passage time of about 30 minutes. For the Japanese white-eye, the times varied greatly from literature, from 12 to 210 minutes. So I chose 3 different time intervals of 30, 60, and 120 minutes. So that's the first part, comparing the distances that they traveled. And next I compared the kernel home range sizes between the 2 species, looking at 50 and 90% kernel home range sizes. Next, to look at the differences in their diets, my collaborators and I first collected fecal samples from birds that were misnetted and captured in bags. And then I looked at the samples under a dissecting microscope. and compared the seeds to a seed bank and also estimated to the nearest 5% what's the composition of the food types in the diet. So looking at some results, after many hours in the field, I obtained data on 9 oma'o, and I found that essentially the oma'o are primarily sticking to inside the kipukas. Each color here on this map represents a different individual that I tracked, and since it's a little bit hard to see, we're going to zoom in on this one area here and show you. So there's 3 individuals represented on this map right now, and the pentagon shapes are the mean center points for this cluster of points, and then the solid lines around the points represent the 50% kernel home range boundaries. The dashed lines represent the 95% kernel home range boundaries of each individual. So once again, as you can see, the oma'o are primarily inside a small kipuka. However, every individual does go outside to the matrix and surrounding areas at some point. Next, I want to show you the locations for the Japanese white-eyes. Again, each bird is represented in a different color, and I do want to point out that for the Japanese white-eyes, they are much smaller than the oma'o, so I had some technical difficulties tracking them using one type of transmitter. The first type of transmitter that I used had some range limitations where the bird was often out of range. So this group here in the southwest corner, I knew in the field that the bird was going farther than I was able to track. Finally, I switched to a different type of transmitter, and this is what I'm going to show you over here, expanded view. This individual in the coral here, I believe, shows most of what the Japanese white-eyes movements are like. So I was able to capture all of this bird's movements using a different type of transmitter. So this bird definitely had a bigger home range than the oma'o, and it spent a lot more time in the matrix compared to the oma'o. So next I want to show some videos of the oma'o and white-eye because the movement is best represented as a time series. So on the left there, you can see that the oma'o is kind of moving deliberately and slowly throughout the kipuka. It does go to the neighboring kipuka in the matrix, but comes back. Contrast that to the Japanese white-eye on the right there, and how it just zooms across the matrix and the kipuka and then comes right back to the same area. So it makes large movements in a short period of time. And I also want to draw your attention to the scale bars at the bottom, that the white-eye is on a much larger scale, so its movements are much greater than this oma'o shown here. Okay, so looking at some results, comparing the distances that the birds travel during gut passage time. For the oma'o, I used 30 minutes period throughout, and for the Japanese white-eye, I used 30, 60, and 120-minute time intervals. So in all instances, the Japanese white-eye, represented in red, had a larger distance that they traveled compared to the oma'o. On average, white-eyes went 170 to 226 meters, and the oma'o traveled 98 meters during their respective times of gut passage. So running a t-test, in all my comparisons, white-eyes moved significantly farther than oma'o did. Next, I compared the kernel home range sizes between the two species, but I found that there was no significant difference in the 50 or 95% kernel home range sizes. And this could be because I only had 9 individuals for each species, and also individuals varied greatly in how large their home range size was. So moving on to diet analysis, looking at the percent composition of food types between my two species, Japanese white-eye ate about 70% insects and about 30% fruit matter. However, the oma'o's diet constituted of almost entirely fruit matter with less than 1% insect. So oma'o had significantly more fruit in their diet than white-eyes. Next, looking at what types of fruits were they actually dispersing. Well, I counted all the seeds that I found in all the fecal samples, and out of 34 white-eye samples, They contained ohelo and akala seeds. However, the 72 oma'o samples had seeds from at least 8 different native species. Just a different way of representing this information. So the graph on the right shows how many fecal samples contain how many seeds. So on the left, Japanese white-eye, most of my samples had no seeds and some had 1 seed in them. And contrasted that to the oma'o on the right. Some samples had 0 or 1 seeds, but many samples had 2 or even 3 different types of plant species contained in 1 fecal sample. So the oma'o not only disperse more seeds, but also seeds of more species. They're much more frugivores than Japanese white-eye are. So in conclusion, the Japanese white-eyes disperse seeds farther than oma'o do, but are much less frugivorous. In fact, they even make larger movements than most of my tracking data was able to show because of some of the range limitations and due to previous studies. So this non-native frugivore in my study site is dissimilar ecologically to the oma'o, and it seems like it won't be able to replace it. This is partially due to the differences in their gape size. The oma'o has a much broader, bigger bill, and it tends to swallow fruits whole. However, the white-eye, even when it does eat fruits, it tends to peck at the fruits and might not necessarily take the seeds with it. This is different from a study on Maui in 2007, which found that the Japanese white-eye disperses seeds from at least 6 different native species, many of which were the same ones found in my study site. I do want to point out that at their study site, they didn't have a native frugivore that was present, so that was one major difference between our studies. In my site, both species are important for primary succession since they take seeds and that they eat inside the kipuka and disperse them outside to the matrix. Therefore, over time, the matrix may slowly regenerate, partially due to the help of these birds. And I want to thank a number of people, particularly my advisor, funders, and Jesse Knowlton, and numerous field assistants for their help in the field. And thank you for your attention.